leaves. I don't know why I keep, buying those, but they're fun. And then my other identity when it comes to plants is being an actual botanist. I got my bachelor's in environmental studies, but now I'm pursuing my doctorate in botany. And so I moved to Wisconsin-Madison to do that. And that's where I study carnivorous plants full time. And so I like to kind of start out by defining what kind of botanist I am, because there's many kinds. But I'm a systematist by training. And so here's a definition I like that says systematics is the scientific study of the kinds and diversity of organisms and of any and all relationships among them. So really, systematics is the diversity of life. And so a lot of or really all the research I do is very taxon specific. That means I focus on an organism, such as the genus Heliamphora, and study everything I can about it. And so taxonomy is one of the fields of science under the umbrella term systematics. And I like this quote as well that says, taxonomy is a synthetic science drawing upon data from such diverse fields, it has no data of its own, kind of emphasizing the point that a fully functioning systematist or taxonomist will use all the tools that it can to get the answers that they're wondering about their organisms. So I put these graphics here because these are a lot of the tools I use personally. Microscopy using a microscope, genetic work like phylogenetics and DNA, and lots of chemical kind of wet lab work. And so most of what I do now is in a lab. I spend a lot of time at a microscope. So if you can see my cursor, this is me at an electron microscope. Sometimes I'm in a wet lab mixing chemicals and extracting plant DNA. Sometimes I'm in an herbarium, which is a collection, like a library of dried dead plants. And sometimes I'm in the field as well. Here I am with some Sarracenia. But I personally kind of prefer the indoor side. And so if you haven't heard the term Sarraceniaceae before, it's a mouthful, but that's the scientific name of the family that houses these three genera, Sarracenia, Heliamphora, and Darlingtonia. And so you guys in California have Darlingtonia. I've yet to see it in the wild, but it's a special one, of course. And so these lines here actually represent the uh, the evolution. So how they evolved over time to become the groups they are. But you don't even really have to think about it like that if you don't want to. You can also think about Sarraceniaceae as just the umbrella term that houses these three genera. And so first, I'm going to talk about my work with one species of Sarracenia, specifically Sarracenia jonesii. And so Sarracenia jonesii grows in the mountains of North Carolina, and it's almost extinct. It's just this well, it's actually not a little species. It can get rather tall. Here you can see that oftentimes the pictures get so long, they fall on top of each other, and it just becomes a kind of big, messy clump. And so it's also in South Carolina, but it grows in these very specific types of bogs and fens that are called cataract bogs and fens. And fen is a habitat similar to a bog. And as I said before, they're very rare and they're federally endangered. They're federally listed, and they're actually almost extinct. And because of this, and also in spite of this, they're very poorly studied. And so, because they have this federal designation as endangered, there are population surveys that have to be done from time to time. And so we can see that there is an overall population decline in Sarracenia jonesii. We know that it's been extirpated from 43% of its known populations. And extirpated means extinct in certain areas. So it no longer exists in 43% of where we knew it used to exist. And so that's really bad. So now it only exists in 12 populations. And frankly, it's been a few years since that number was calculated, and it might even be less. And so 12 populations is absolutely not enough because we need more and more populations to increase the genetic diversity. And so I studied one of those 12 populations called Robert's Nature Preserve. And in this population, there were three subpopulations. And so this is kind of like a fake map I'm making to kind of put it in your brain. And there used to be a fourth subpopulation at this nature preserve, but it was last seen in 2018. And myself and multiple botanists tried to find it, but we could not find it at all. So I think it has also been extirpated, meaning it no longer exists. But overall, this preserve is called a metapopulation, and these little guys are called subpopulations, except this quote unquote subpopulation was really just one individual all the way by itself. And so, for the purpose of my study, I defined a clump as a group of individuals distinctly grouped together, likely sharing rhizomes. So, here's an example of me working with a clump of Sarracenia jonesii. And if you've ever, unpotted a Sarracenia, you might be familiar that sometimes in one pot you could have multiple individuals. So in theory, likely if this was dug up, there would be multiple individuals that are sharing rhizomes. But for my purposes, this is one clump as I defined it. And so let's quickly go over some of the overall morphology or parts of a Sarracenia plant. We know the very famous pitcher, the carnivorous tubular leaves, then the less well-known non-carnivorous leaves, which are called phyllodia or phyllotium in the singular, and then, of course, the flowers. And for my study, it didn't matter if the flowers were fresh and beautiful or if they were dried. And so I had four main hypotheses, and I'll introduce the hypotheses and then I'll go into each one more specifically. But I was hypothesizing that the more non-carnivorous leaves or the more phyllodia would be found in the clumps that have the most plant available nutrients in the soil. And I'll keep throwing that term around, plant available nutrients, but I'll explain that more in a little bit. So that was the first hypothesis. The second one was that the soil in which Sarracenia jonesii grows in is not nutrient poor, but rather nutrient rich, meaning a high total nitrogen. But these nutrients would not be in plant available forms. Again, I'll get into that terminology. Third, I hypothesized that the more plant available nutrients in the soil, the fewer the flowers the clumps would produce, which kind of might seem contradictory. And then my fourth hypothesis was that as one of the last remaining or extant sites, I hypothesized that the overall meta population at the nature preserve would have declining trends in population since 1979. And that is the date chosen because that's the earliest population survey available. And so I won't bore you with my lab methods or my statistical methods, but I'll show you some of the field work I did, where at each subpopulation, I randomly selected 10 clumps to survey. And at each clump, I counted the number of pitchers, which is, in the thousands. It was a lot of counting. The number of phyllodia or non-carnivorous leaves, the number of flowers, the clump circumference, and then I also took these soil cores so that I could get samples of the soil that the clumps were growing in. And so going back to the hypotheses and seeing what I found for each one, for the phyllodia hypothesis, I thought that the amount of plant available nutrients would affect phyllotium growth, but that's not what we saw. I built this hypothesis based off of the scientific literature showing that the more kind of nutrients you feed a Sarracenia, the fewer pitchers it would grow and the more non-carnivorous leaves it would grow. And I thought this because in the sense that if the plant is getting enough nutrients from the soil, it would no longer need to catch as many prey. And the payoff there is that carnivorous leaves, like pitchers, are very expensive to make. It takes a lot of energy from the plant. So if they don't have to make them and then can instead make these non-carnivorous leaves, I would think it would benefit them. But I didn't find that relationship. And it's likely that I think is that these studies showing that relationship, in cultivation we're using higher rates of nutrients, such as actually fertilizing the plant, than we'd see in a wild environment, in at least a healthy environment. So there are a few graphs, but you really don't have to read them because I'm explaining it with my words. I know who wants to read a graph on a Saturday, but this is just showing that there isn't a strong relationship. And I also looked at if there's a relationship between the number of plant available nutrients and actual pitcher growth. And again, I did not find that relationship. But going to this hypothesis, which is actually what started this whole project, I know that I'll back up. So we always describe carnivorous plant soils as nutrient poor. I feel like if you have any carnivorous plant book, or if you even read the little name tags at a plant nursery, the soil that carnivorous plants grow in are always described as nutrient poor. But part of this project is I wanted to push back against that terminology because I do believe, and I'll show you some of the data, that that's not entirely accurate. So when we say nutrient poor, what that really means is nutrient unavailable, which might sound like semantics, and it is a little bit, but to scientists, it's not. It is biologically and chemically different. So processes that turn certain nutrients from plant available to plant unavailable is mainly done through decomposition. And so chemically, some compounds cannot be as easily taken up by the plant. So those would be called plant unavailable. The plant can't take them up chemically. But then some are easier for the plant to take up, and those would be called plant available. And decomposition is the process by which unavailable compounds can become available. So if you think about, if you're composting your food waste as pure compost, undecomposed, a plant couldn't take up those nutrients. But once it's decomposed into that kind of soily mush, then those nutrients are likely in a plant available form. But decomposition doesn't really happen that much in soils that are super wet for a long time, and so meaning not a lot of oxygen, and in very acidic soils. And both of those descriptors of being very wet and being very acidic describe a lot of the soils that carnivorous plants grow in. And those values don't contribute to decomposition, which would then likely not contribute to making plant available nutrients. That was a lot of words, but hopefully it kind of made some sense. But I'll explain here on the slide again. So I actually found, supporting my hypothesis, that the soils had a lot of nutrients, but they just were not plant available. And so you don't really have to read the graph, but here in this table, I'm comparing what would be a normal soil, aka a soil that a carnivorous plant would not grow in, like just an average soil, and versus what the Sarracenia jonesii was growing in. So a normal soil would have about 0.1% total nitrogen, and ours had zero point 0.2. That's a very big difference. And these are the plant available forms of nitrogen of 2 to 10 parts per million in a normal soil, but on ours it was 0.5. That's much smaller than even the smallest number here. And again, here we see a much smaller amount of NO3, which is a plant available form, than in normal soils. And so again, this supports the hypothesis that the fen soil is actually not nutrient poor, but it's nutrient rich. It has lots of nutrients that have not yet been decomposed and turned into plant available forms. So when folks say that soil that carnivorous plants grow in is nutrient poor, they really mean it's nutrient unavailable because it's higher in nutrients than an average soil, but those nutrients can't be taken up by the plant. So it's kind of functionally nutrient poor. So these are just some quotations from scientific papers calling these soils nutrient poor. So if there are more studies like mine looking at other carnivorous plants, looking at other habitats that can show the same relationship, because I hypothesize this would be found in all carnivorous plant habitats essentially, hopefully we can start to move away from that term of nutrient poor since it doesn't seem to be very chemically or biologically accurate. And then for the flower hypothesis, I found that the number of flowers grown and the amount of NH4 in the soil did not have a statistically significant relationship. But I did find that the more pitchers a Sarracenia jonesii clump grew, the more flowers it grew. And theoretically, that also makes sense because if there's more pitchers, maybe it eats more bugs and then it has more energy to flower more. And then for the population hypothesis, as I thought, the metapopulation is slightly declining in the number of clumps over time from 1979 to 2022. But the clumps themselves actually seem to be growing more leaves and more flowers in the past, even though there are fewer clumps. And that could be due to a few things. Possibly because these clumps are older, they're multiple decades older now, and the nature preserve now knows these plants are here and are trying to increase the conservation efforts. And so hopefully the goal of this paper was to provide more information about the species overall and this habitat overall, and it can be used to inform conservation strategies when scientists and hobbyists alike grow this plant in cultivation. And it was just kind of further evidence that this habitat needs to be protected. And with more similar studies regarding the carnivorous plant soils, hopefully we can start to move away from that nutrient poor label and switch to plant unavailable. And so this was finally published as my first author paper this year, if you're interested in learning even more about that work. And then, so I showed you the family already, and I just talked about Sarracenia. But now for my PhD, which I've done for the past 2.5 years, I'm working on Heliamphora. And so I'll jump into my Heliamphora work. And so the title of this part of the presentation is that scanning electron microscopy, or SEM, reveals species-specific epidermal traits in the carnivorous genus Heliamphora. So I've already introduced the family to you, but here are three pretty pictures of each genus. And actually, Heliamphora is the most species-rich genus in the family, with about 24 taxa. And it's most well known for its nectar spoon, which is this piece of specialized tissue that has a high density of extrafloral nectaries. And it's different from the lids that we see in Sarracenia, by the way. They have different purposes, but it makes it easy to identify a Heliamphora specimen. And because I know we like to grow plants here, I thought I would mention their common name for a second. So it's often called sun pitchers or Mars pitchers or South American pitcher plants. And for scientific names such as Heliamphora, there are so many rules. Every 5 years, botanists of the world come together and vote on new rules, and there's a new handbook. So they met in Madrid last year, and this is the new rule book to follow. But for common names, such as these three in the corner, there are no rules. There are no biological standards. They're common names for folks to be able to use colloquially. So really, there's no right or wrong one because there are no rules. But if you wanted to try to be the most proper, when Heliamphora was described by a botanist named George Bentham in 1840, he named them Heliamphora, and he said it was for being called a marsh pitcher. People think Helia means sun, which it does. If you think about sunflower, its scientific name is Helianthus, and that Helia meaning sun, but he meant Helia as in marsh. And so that's just and he amphora because it's like a pitcher like this because it resembles the shape of a leaf. But again, there are no rules for common names. And so Heliamphora is the only one in the family that's native to South America. And it was discovered in 1829, but is still one of the least understood groups of carnivorous plants overall, not just pitcher plants. It's just very not well understood. And that's largely due to its inaccessibility. For example, they grow on the tops, sides, and bases of these very special mountains called tepuis in northern South America that are these tabletop mountains that go straight down all around, and they're very hard to access. And so here is a view of the whole genus just at a glance. It's a lot of pictures, I know, but I'm showing you all these pictures to kind of show off the idea that I think they're very similar looking macromorphologically, meaning if I just take a step back, they all kind of look like these red or green tubes, right? And so when I started out my PhD, I wanted to try and figure out a way to differentiate them even further. And so one of the ways I'm doing that is through their trichomes. And trichomes are just the botanical name for plant hairs. They are specialized epidermal cells. And in my opinion, they are integral to the identity of Heliamphora. They're used for their taxonomy, how you tell the species apart. They're used for how you identify different species. And they're also essential for their prey capture. And in fact, 5 of the 24 species are named for their trichome characteristics. And so for my research, I use samples purely from horticultural collections, so there's no field work. And here are some photos of the collections. And so I collected leaves from 52 different accessions, which is like 52 different localities, specimens taken from different locations. And these together represent all 24 species. I also used some pressed specimens that came from the wild. Those are called herbarium specimens. And I used multiple hybrids and potential new species as well. And so I'm going to throw these terms around. So I wanted to introduce them if you're not yet familiar. Adaxial and abaxial. Adaxial is what is called the top side of the leaf. And then the abaxial is the back side or bottom side of a leaf. And you can differentiate those because abaxial has a B for backside or bottom. So I wanted to see really up close, are these leaves different between each other? Can I find differences between all the species once I take a really close look? And spoiler alert, I can. So let me show you how I went about that. I took each leaf that I had grabbed from all those different specimens, and I created these 4 different zones that I considered to be taxonomically important. And from each zone, I cut off a little piece of tissue that I would then put under a microscope. But I couldn't quite yet put it under a microscope. I had to first put that tissue in this instrument called a critical point dryer, which dried it without wrinkling it. So, for example, if you put a grape in that instrument, a grape would come out not wrinkled. It would be perfectly spherical, but completely dry. It's really cool. And then I would put it into an instrument called a sputter coater, which would coat your sample with platinum palladium to protect it. And then finally, I could put it into an electron microscope, which uses electrons instead of photons, light, to view your samples way, way, way up close. And so I'm going to just show you what I generally see on a Heliamphora leaf up close. And I'm going to be using one species as an example, Heliamphora chimantensis. So when I look at that nectar spoon up close, I, with no surprise, see a lot of these extrafloral nectar glands. And they're very similar to the ones that we see in Sarracenia. They're very large. And I don't tend to see a lot of trichomes or those plant hairs in this area. When I look at the top inside of this leaf, which here I have the adaxial, which is the inside of the leaf, and the abaxial, which is the outside of the picture, I see a lot of trichomes of different sizes. They're lined with these ridges. Sometimes they have these cells around the base of their trichomes that create a sort of rosette. And then on the back side, I see a lot of double-sided or bifurcated trichomes and a lot of stomata, which are the pores of a plant, which are these little slits. And then in the middle of the leaf, I see even longer trichomes, and sometimes they swoop upwards, sometimes they hook downwards. And again, lots of different sizes. I see little ones and really big ones. And then on the back side, again, I see double-sided trichomes. They're kind of like 180 from each other, the two sides and floral gland or nectar glands and more plant pores, stomata. And then in the bottom, I typically see these long, straight trichomes without ridges. And on the back side, sometimes I will see trichomes, and they're usually double-sided, and still stomata and nectar glands. And so that was just one species as an example. I did that for all species and multiple samples per species, and I found lots of differences. So, for example, we're looking at the same area in a leaf, the top inside, but in three different species, and we're seeing some of these big differences. In this species, it's hairless, right? This species, it has a good amount of trichomes, and in this species, it's way hairier. And then here again, same spot in a leaf, three different species. Some we see these kind of intermediate length trichomes. Some we see these are kind of short and squat, and these are really long and thin. So before so that I can kind of try and differentiate the species. Again, same spot in a leaf, three different leaves. This one has a little bit curly or wavy trichomes that are double-sided. These are very straight, and these are just non-existent. And so it's really cool to be able to know that once we look at these under the microscope, I can try and tell them apart now. And then I went even further, focusing on one species in particular, Heliamphora pulchella. If you're into growing Heliamphora, you know this is a very highly sought after species. It can be rather expensive. And it's known as a species complex, where what we consider Heliamphora pulchella may contain actually multiple species because it can be very plastic, meaning it can change a lot depending on how you grow it. And so I wanted to dive into that one a little bit deeper because we know there are some weird things going on. And so, particularly, the hairiness or these trichomes differ a lot. So, this is one specimen from one Tepui mountaintop that's rather hairy, and this one that has just a few hairs. And so I had an undergraduate student of mine, he's now graduated, but he did a senior thesis with me where we looked at the species really up close. Together, we used 13 accessions or these localities of Helianthera pulchella. We all got the specimens from cultivation, but originally they were from, or they came from plants that came from these various Tepui mountaintops. And so I'm just going to show you a few examples of what we typically are seeing in these Helianthera pulchellas. So this is an example, or these are all 13 of the accessions we looked at, and these are images from the top inside of the leaf. And immediately I can start to see some differences. For example, some specimens have really long trichomes with lots of little ones growing around it. Some have just more intermediate sized trichomes spread throughout with smaller ones spread around it. Some lack these longer trichomes altogether. And so it's up to the student and I to, as biologists, as taxonomists, figure out, is this actually significant in telling us what is Helianthera pulchella and maybe what's not that species? And so that's kind of all I had time for. But I first, before I end, I wanted to thank, of course, the various students who helped me, my pitcher pals. They're a group of undergraduates, and my advisor, of course, and all these other folks and these other funding sources that have made it possible for me to do my work, including you guys, the wonderful San Diego Carnivorous Plant Society donated to my research as well, and that has been essential. I'm almost able to now afford my whole next new project on Helianthera, and I thank you greatly for that. And I also just wanted to end with this remark that I don't think I could talk about science right now without mentioning the fact that, science is in danger. And this is a nice article if you're interested. It came out a little over a year ago now on the fact that botany seems to be dying. It's endangered. There are fewer botanists being trained in the United States, and funding is an all-time low for botany. And it's a really fascinating article. But I just kind of wanted to remind everyone that scientific support is essential for society and mitigating climate change. And even studying obscure little pitcher plants like I'm doing now is important for understanding life on Earth and our place in it. So I just wanted to remind everyone to stay curious and stay engaged with the natural world and scientists and scientific world in general. So that's all I had to share with you. And so if you have questions, I'm happy to take those, but this is also my social media. I post a lot on here. You're welcome to email me. I do educational videos on Instagram. I do a little bit of TikTok and Blue Sky, but I'm mainly on Instagram.
Great, thank you. And I think you have some time for some Q&A if people have any questions.
So,
So that's the mic so that he can hear you.
oh, okay. So, did you are you going to be going into some more taxonomic work with Pulchella to see if you can identify actual different species in what we consider as one?
That's kind of the point now that I'm at is to start that taxonomic work to see if there are differences. I didn't enter the project thinking I would split it apart, and I wouldn't do it purely myself. I'd do it with collaborators, but I can't say yet. I don't know.
Yeah, because I grow the Acapen Pulchella and it has almost no hair.
Right? Yeah, yeah. There's quite a bit of difference between the
Yeah.
specimens. But, the hard part for taxonomists is figuring out, is that significant enough to mean it's a different taxon, or is that
Mm,
just variation?
okay. Thank you.
Yeah, thank you.
I'll follow you on Instagram. I appreciate the work that you're doing.
Thank you.
So my question is mostly with the Heliamphora. So it's interesting that they grow in a much more harsh environment than the traditional Sarraceniae and Darlingtonia, but they only occupy a much smaller region. Any idea why there might be more species there versus stateside here?
That is a beautiful question that everyone wants to answer. We aren't exactly sure why. It could be as kind of a side effect of the habitat that they grow in, where they are very disjunct from each other. The Tepui mountaintops, there's not a lot of, it's not very easy to transfer between them, so it can isolate populations or specimens very easily. And when populations are isolated, it's easier for their kind of genetics to differentiate from other populations nearby, which leads into speciation or different species, if that makes sense.
Yeah,
Good
thank you.
question.
Yeah, thanks for the great talk. So I'm curious about the three genera that you mentioned, Darlingtonia, Sarracenia, and Heliamphora. So they are like very separated geographically, right? So I'm wondering
Yes.
whether you know or like know any hypothesis of how they got separated, or were they like kind of a convergent evolution to become carnivorous like separately?
Yeah, that's a great question, and one that all carnivorous plant scientists are wondering. We aren't exactly sure yet. They're very young evolutionarily, meaning in the history of all of Earth, they evolved very recently. And so we don't have any fossils of them. So we don't know exactly how they came to be. And actually, one of the family's closest living relatives is the kiwi plant. And so there are a couple functioning hypotheses, some being that they started in South America and then moved to North America when the continents separated, or they could have started in North America and then Heliamphora came down. But now that we know that Heliamphora and Sarracenia are more closely related to each other than to Darlingtonia, it seems like maybe the Darlingtonia was the one that went off on its own, and Heliamphora and Sarracenia started together. So I don't have a perfect answer because we don't really know.
Yeah, thanks.
Yeah. But those are awesome questions. Thank you so much.
Hi, I was just wondering if you do any genomic analysis on these and if there's any kind of diversification, like let's say the Sarracenia, like subpopulations, and you see any potential, um, I guess, diversifying enough and there like might be speciation.
Like in Sarracenia in general.
Yeah, like the one, well, no, the one that you were studying in that
Oh,
locale.
I see. Yeah. No, so I didn't do any genetic analysis on Sarracenia jonesii, but there are groups of people that are doing that. I am focusing now on doing genetic analysis of just Heliamphora, but some of the preliminary genetic analyses of Sarracenia jonesii suggests that it's the species most at risk for extinction in the whole genus. And it's showing that it's hybridizing way too much. So it's actually not really speciating or turning into something else. It's being lost to its hybrids. Its genetic diversity is getting muddled, actually. If that answers your question.
Any other questions?
Well, thank you so much for having me and
Yeah, one
donating
more round of
research.
applause for Joseph. Thank you again so much for making the time to present and share your findings with us. Yeah, if no one has any other questions, we can end it here. Hopefully,
Okay, well, thank you
your
so
day.
much. Have
All
a
right.
great rest of your Saturday.
Thank you. Thank you.
Okay, bye.